Hello. First of all, thanks a lot to the organizers for having me here. It's a real honor to be in such a distinguished circle of researchers. Yeah, I'd like to talk about human data, but of a more invasive version, namely single neuron recordings that we perform in epilepsy patients. And the data I'll be showing today is all from the human medial temporal lobe. So I'd like to first describe how we actually are able to record these signals and then describe the, to me, very seminal finding of so-called concept neurons and what their potential role might be, especially in declarative memory formation. So I guess most people would agree that the basic informational unit of the human brain is the neuron. And fortunately or unfortunately, we have around 80 billion of those. So that's quite a large number, and it seems not feasible to characterize all of them at once. And especially for human cognitive research, it's really invasive to get close enough to a neuron to record from it. That is why non-invasive techniques have been used, like fMRI, as we have just seen in Stan's talk, also MEG, EEG. And all of these, well, fMRI has the issue of a poor, limited temporal resolution. The spatial resolution of all three techniques is limited. The big advantage is that they cover the entire brain. And then on the other side, we have lots of microscopic knowledge from in vitro recordings from human tissue. And what is basically missing is in vivo electrophysiology at the single neuron level, which is why there's a huge amount of research going on in animal models, such as rodents or non-human primates, not primarily because we're so keen on learning about the rat's brain, but because it seems like a decent model or hopefully a good model for example, for memory in the human brain. So we perform this type of invasive research in animals in order to, or with in hopes of better understanding the human brain. Now, the data I'm showing is recorded from epilepsy patients. There's, roughly speaking, two types of epilepsy: genetic epilepsy and the structural or focal epilepsy. And in focal epilepsy, usually there is some alteration of gray and white matter in the human brain. There is a focus where the seizures start, and many of these patients do not become seizure-free taking medication. And in that case, their best hope of becoming seizure-free is to actually identify the seizure focus and to neurosurgically remove it. And then ideally, the patient will be seizure-free and have no cognitive or neurological deficits. Now, in the absolute majority of cases, roughly 90%, this can be done non-invasively using scalp EEG, surface EEG. But in maybe 10% or less of patients, it's impossible to identify the seizure-generating brain tissue in that way. And for those cases, if they want to know if they are a candidate for surgical removal of the epileptic focus, we need to record the seizures invasively where they start. And in that case, the patients can choose if they want to go on. We need to implant electrodes into their brains, and electrodes like the ones pictures in the schematic there have been around for at least 50 years, so-called depth electrodes, and they have cylindrical contacts that allow to record the electrical activity of bulk brain tissue, maybe a milliliter of brain volume there. And then maybe some 20, 25 years ago, a neurosurgeon by the name of Itzhak Fried at UCLA had the idea of using the fact that these electrodes typically have a hollow inner canal where there's a little steel fillet membrane that makes them more rigid, to simply use this inner canal and feed a bunch of little wires through the electrodes once they're placed in the brain so that they stick out at the end, maybe 4 millimeters. And these wires are small enough that they record a volume that in turn is so small that only one or a handful of neurons are being recorded. And this allows us to record action potentials of single neurons in awake, living, and behaving humans. The upper image here shows you CT and X-ray reconstruction of implanted electrodes. Electrodes are typically implanted orthogonally from both sides. Often something like 5 to 6 depth electrodes per side. And if each of these electrodes have a microwire bundle, these bundles typically consist of 9 wires, 8 of them to record action potentials, and another one with stripped insulation at low impedance to be used as a local reference. On the lower right, you see an MRI, structural MRI reconstruction. The electrode contacts here appear much larger than they are due to susceptibility artifacts. This is an image from our OR. This is how we implant these electrodes. So on the left, you see the macro electrode being implanted, and the little stillet inside of it is then being pulled out. And then I typically feed those microwire bundles through the electrode. We don't record in the OR, so we don't know what we get till the next day when we start recording. The data I'll be showing today is from the medial temporal lobe, and our standard implantation scheme at the University of Bonn is to, on each side, have 2 electrodes in the hippocampus, 1 in the amygdala, and then 2 in the parahippocampal gyrus, 1 so far anterior that it hits the entorhinal cortex, and the other so far posterior that it'll hit parahippocampal neocortex. Here's a picture of one of our epilepsy monitoring unit rooms. So you see the patient here with a big head dressing because we have to do the preamplifiers. They need to be fit into the head dressing. We need to preamplify the signal as close to the electrode as possible. It wouldn't be possible to transport it across the room without getting totally contaminated. The patients typically are at the EMU for 1 to 2 weeks. During that time, they wait for their seizures to occur, and they're not allowed to leave the bed, so they're typically quite bored and also quite motivated to participate in cognitive experiments. Now, in a good patient, where we have a good yield of units using these 10 depth electrodes, meaning 80 microwires, 80 monotrodes, after spike sorting, we get about 100 to 150 single and multi-units. When a unit looks nice, we call it single unit, the rest is called multi-unit. This is more of an operational definition. Anyway, we get to record 100 units from out of 80 billion neurons across different areas. So in a way, you could call this whole project hopeless because we're hopelessly undersampling the system in terms of numbers. And so we can ask, what can we expect to learn in the first place? And this relates to a discussion that was led among monkey electrophysiologists in the 1970s. So how can our environment be represented at the neuronal level? And there are 2 extreme ways of coding. One is called distributed code. And just as an example here, I have a network of 8 units. And if we add the constraint that half of them are always active, half are not, there's only on and off, then this would allow us to distinguish 70 different states. It's just the binomial coefficient 8/4. So by feeding only 8 units, we can distinguish 70, say, people or objects. Another way of coding, another extreme way would be a sparse code, where we simply have 70 neurons to represent, say, 70 people. And this one black neuron, for instance, could represent your grandmother, which means it would fire whenever you see your grandmother, whenever you talk to her on the phone, whenever somebody mentions her name, whenever you think of her. It's the idea of the grandmother coding, and it's often mentioned in neuroscience textbooks as a way the brain does not work, because obviously it would be way more efficient and economic to use a distributed coding than such a sparse coding. So this is something that's supposed not to be present in the brain, but I think the findings of the last 10, 20 years argue strongly against this, and I'll be showing you a movie now where you get to listen to a neuron while the subject, the owner of the neuron, is viewing different pictures. I hope the sound works. Every click is an action potential. So this is a neuron that has a clear preference. for one of the pictures. So as you can tell, there's some background activity or baseline activity, and then whenever the picture of Marilyn Monroe comes on, the neuron goes crazy. This is something that behavior that we observe in virtually every patient we tested with. And what you see here is a visual depiction of what you just listened to. So here we have the target stimulus on top, and then you see these 6 raster plots with the blue lines. Each blue line represents an action potential, a click that you just heard. And below that, we have a histogram where we sum up that activity. So the x-axis contains 3 seconds worth of data, and the dashed lines are onset and offset of the picture. So the picture comes on, and then 200-300 milliseconds later, the cell starts firing, and then it stops it. Now, the data I just chose you is something that we recorded at UCLA a couple of years ago. These days, I work in Bonn, and we don't have so many Hollywood celebrities that we get response to. But this is Michael Ballack, a former German soccer player, and we had a patient who responded to a picture of his. The general question with these selective responses is, what is it that drives the cell? And there's a huge number of potential explanations. So if you take the Marilyn Monroe cell, it could be a response to women, to blonde people, to white dresses, or to black and white pictures. But of course, it could also simply be the identity of the person. And it turns out that that's what it is. So here with the Michael Ballack cell, we ran a follow-up test where we showed different pictures and of people, their written names, and also their names spoken by computer voice. And it turned out that the cell didn't care for any of the other stimuli except for Michael Ballack's picture, his written name, and his name spoken by a computer voice. Now, the first 2 are visual modalities, although language and names and words are processed very differently from faces. And the third one is an auditory stimulus, which has a completely different processing pathway. But all of this converges in a hippocampal neuron that is super selective. And we know, I don't have data here to show it, we know that these cells are also activated by imagery. So as soon as this patient thinks of Michael Ballack, this neuron will fire super reliably. The next picture shows a different example. Here we had, in the morning testing, we found a cell that was responsive to Harry Potter. And in the follow-up experiment, my graduate student made a mistake and clicked on the picture next to Harry Potter, which happened to be Ron Weasley. Ron Weasley is Harry's best friend in the Harry Potter saga. So this unit responded to Ron Weasley and to the written and spoken name of Harry Potter. We know it also responded to the picture of Harry Potter earlier on. I would be very confident that it would fire in response to the written and spoken name of Ron Weasley. If you're familiar with the story, there's Hermione Granger, who's the third of a bunch of friends there. I would guess it might respond to that as well. Then I don't know if it might also respond to Lord Voldemort or Professor Dumbledore, or maybe even to Gandalf from Lord of the Rings. The reason I'm showing this paper, this response, this slide, is to drive home the point that these cells have what one could call a semantic tuning curve. And we find very narrow tuning, and we find wider tuning, and our impression so far is that basically any and every kind of tuning is present in the medial temporal lobe. This is some ongoing work in collaboration with Stan, and so much right now for these cells. There's a couple of other features, but I don't have time to go into that. The next question would be, why would the brain allow itself the luxury of having these very sparse representations when we've just realized that it's not super economic or anything of that kind? And to get to the bottom of that question, first of all, it's useful to ask, one important feature is we find these cells exclusively in the medial temporal lobe. To my knowledge, they have not been observed elsewhere, and also they have not been observed in this way anywhere in the animal kingdom. There is a recent paper on marmosets, but I believe I have strong arguments that these are very different cells. Anyway, so we know that the medial temporal lobe is important for memory, and we know that because there is this, one of the most famous case studies in cognitive neuroscience is patient HM, who was the first victim, one has to say, of epilepsy surgery in the 1950s when both of his medial temporal lobes were resected. And it turned out that for a while he was seizure-free. He had a bit of a retrograde amnesia, but the most important thing was he had a complete anterograde amnesia, which means he lived on for over 50 years, and he could never remember or later recall any of his experience from the moment of surgery on. So even in his high age, he could remember things from his childhood, stuff like that. He could not remember a single thing. Since the surgery, he had caretakers who would come to him every day for 10 years, and he had no idea if he had ever seen them before. Another important feature is when we look at the response onset latency. And one nice thing about electrophysiology is that we have a good temporal resolution. We see that these cells respond very late, roughly 400 milliseconds after stimulus onset, which is much more time than the human brain needs for object recognition. So object recognition has long happened by the time these cells start to fire. We also know that conscious perception is required for these cells to be activated, right? If I can trick you into seeing something that's not there, these cells will also be tricked. So they strongly correlate with the conscious perception, meaning that the emergence of a conscious percept must have happened before these cells get activated. And maybe the earlier regions, such as the parahippocampal cortex, subserve object recognition and are where conscious perception is initiated. Anyway, the last thing that I didn't show you for lack of time is that indeed they follow the conscious perception. So there's actually 4 different studies by different groups showing this. For now, you just have to take my word on it. But if we realize, we know episodic memory encoding is what these regions do, and memory happens after object recognition, and declarative memory by definition requires conscious perception, then it's not hard to come up with a hypothesis that maybe these neurons simply provide the semantic building blocks that are being associated together, pieced together to form an episodic memory. So if we go out to dinner with some friends from work and suddenly we run into an old kindergarten friend, right? Then that is something that we need to be able to remember the next day, even though the people there, the persons are not semantically related or hierarchically related in any way. So the medial temporal lobe needs to be able to arbitrarily associate concepts that we can consciously recognize. And in order to do that, we need to have sparse representation, because a network can do all kinds of things, but it can only do one thing at a time. And if we accept that episodic memory can be operationalized as associative memory, and associating things at the neural level means firing together, then I believe that this concept first proposed by Rodrigo Quiroga makes a lot of sense. And we are the first ones to, I think, report actual evidence for these findings. From now on, this is unpublished data. This is a task, an associative memory task, where in the morning we run a screening. We show all kinds of pictures of objects or of people, typically customized to the patients, to their hobbies and likings, because we know the more familiar and or relevant pictures that we show are, the higher our chances of finding selective responses. So we use now, after a screening, the response eliciting pictures for a follow-up task, in which we have a 3x3 grid, and a series of images is shown at different positions on this grid. So in this first example, 3 different pictures. The patients have to touch the pictures on a touch screen, because at the time we did not yet have eye tracking integrated. Then there's a distraction task of 15 seconds. And then there's a recall task where the same series of pictures, but in random order, is presented with a picture below the grid, and the patients have to indicate where the picture was learned on the grid by touching again. And then we run an adaptive algorithm to make sure the task is difficult enough, because ideally we want to have 50% successful, 50% unsuccessful trial. And in order to get closer to that, The better the patient get, the harder we make the task by adding or leaving out one of the pictures in this series. So that is the paradigm. And what we find, I'll skip the single unit examples, but at the population level, we ran this in 13 patients, and we recorded a total of more than 3,500 units. And this is the grand average of the instantaneous firing rates. And here in the first column, you see the item neurons, the ones that really showed selective responses, which, as you can tell, is less than 10%. So in the amygdala, we have 80 item neurons out of, I think that's 1,117. I think I need new glasses. Anyway, and we find that their firing rate during encoding allows us to predict whether there was successful memory formation or not, right? So the blue are the successfully remembered ones, the red are the forgotten ones, and we see that it is the activity of the item neurons that allows us to predict whether successful memory will be formed in the three regions, amygdala, hippocampus, and entorhinal cortex, but not in parahippocampal cortex. Now, we also found location neuron cells that responded selectively to one of the 9 fields in our 3x3 grids. Actually, the proportion of them using a proper statistical permutation test was insignificant in the first 3 regions, but it was significant in the parahippocampal cortex. And here we again found that firing activity predicted memory formation, but at a very, very late stage. We're still surprised by that. We ran all kinds of controls and it just wouldn't go away. So we have to report it. Here, for example, now we align everything to the bottom press, right? To the press rather than the stimulus. The effect is still there. We can't get rid of it. But maybe most importantly, here now we have the 90% other neurons that did not respond selectively either to a picture or to a location, and they do not predict memory at all. So even though now we have tenfold the statistical power, we don't really see an effect. The y-axis is scaled in the same way. The little effect we see in entorhinal cortex, I would argue, is false positive or due to our very strict response criteria. Those are semi-responsive cells. So really, it's specifically these concept neurons that allow us to predict successful memory formation. Now, this was a correlative study. Of course, it would also be nice to interfere a little bit with memory. And this is an old study from some anesthesiologists that investigated certain sedative drugs and narcotics and found that propofol has a very, very strong amnestic effect on patients. So more than any other drug investigated here, the red dots are propofol. Even at low target concentration, the recall in a picture learning task was strongly diminished, which basically means propofol leads to an anterograde amnesia, where we have a dissociation of consciousness and memory. We consciously perceive things, but we're not able to remember afterwards. And it turns out that in our patients, when we explant the electrodes, we use a brief propofol anesthesia. And because of that, it wasn't hard to convince our ethics committee to approve this study and allow us to start the propofol a little more slowly, run a cognitive task, and then proceed with the surgical procedure. And we did that only in patients where we had a very good, trustful relationship, and patients who had good units and good responses. And so this is 9 sessions, 9 explantation sessions, but more than 500 units. And the design is a bit complex, but I'll walk you through it. So we start in the morning doing our screening session. We pick the 8 most promising response eliciting pictures and another 16 control pictures. Then we move the patient into the OR. In the OR, first we simply run a mini screening to see whether these 8 responses are still there, and we also add 8 of the control stimuli. The patients simply see the pictures and have to name what they see. Every picture 12 times. Then we start the propofol up to a target concentration, estimated target concentration of 1.5 micrograms per milliliter. The patients are usually feel a little tipsy. Some of them get very talkative. They're usually in a good mood. And now we run a task similar to the previous one, where first they see, now we only take the 8 pictures that we're actually interested in. First they see the pictures, they have to name it again. They're under propofol, but they're able to recognize what they see. They're, in that sense, perfectly conscious. And then they see the picture on a grid. And this time, every picture is always at the same position in the grid. And they have to memorize the position. So 12 times they see the picture, they name it, they acknowledge where it is. And normally this would be a piece of cake to learn for them. But it turns out it's hard enough. So as soon as we're done with that, we stop the propofol, we wait till the estimated target concentration drops below a certain level, and then we have 3 recall phases. So in part 1, we simply ask them, we now add the 8 pictures that we haven't used yet in order to figure out whether in the mini screening, the green task, whether they still remember what they saw there. Next thing we do is we ask if they know which of the 8 stimuli were in the grid task. And then finally, we ask for those 8 stimuli if they remember the position. And then we repeat the same thing the next day when the electrodes are already gone to have a control. And it turns out that subjects reliably remember what they saw in the green task, in the first naming task, but they're already at chance performance, more or less, or strongly decreased, to remember which of the stimuli were in the grid task, something that has to do with familiarity, right? And when they're asked to report the position of the pictures that they've learned, which is what one could call source memory, they're quite, right? Now we can compare the electrophysiological dynamics for remembered and forgotten stimuli, and this is what's shown here. Relative to the mini screen, to the baseline, we see what the decrease, what the attenuation is in firing. So in this case, it's the peak firing rate when the image is shown during the propofol task. And we see that there's a significant effect for this item memory effect in hippocampus and amygdala that allows us to predict whether things will be remembered or not, but not in parahippocampal cortex. And for the source memory, for the stimuli where they actually remembered the position, we find the effect only in the hippocampus, which aligns well with the literature on hippocampus. Next, we looked at the attenuation on the grid, because only then all the information is present that they need. And we find the same effect, basically. And finally, we looked at the delay of the onset of the peak latency. Now we have to go back again to the first presentation, because once it's on the grid, we no longer observe the peak. And we find that this delay is shown only for source memory and only in the hippocampus. So in this case, we can use a pharmacological manipulation to alter the firing dynamics and see that this directly influences memory. And with that, I'd like to close. So we're very lucky to find these very sparse representation, because otherwise we would, I think, be hopelessly undersampling any network code. But with these concept neurons, we can look what their meaning is. We see that they clearly are a correlate of conscious perception. It makes sense to have them in this sparse fashion so that they can be freely associated. And we see that it predicts successful memory formation and that we can pharmacologically perturb this. So we think this is the first direct evidence that concept cells are indeed providing the semantic bits of information that is being pieced together to form episodic memories. And with that, I'd like to thank my collaborators, especially for these two studies, people from my group, and you for your attention.